Yeah. Thank you, and thank you to the organizers for inviting me. I'm very thrilled to be able to speak here. Now, my title is Signal P6, Enhancing Signal Peptide Prediction with Protein Language Models. But I could also have named my talk, What Can Language Models Do for Protein Science? Because that is what I'm going to show you. So in the section for bioinformatics at the Technical University of Denmark, we have several web servers that predict various features from either nucleotide or amino acid sequences. And some of these are based on protein language models. I can mention Signal P6, which is for signal peptides and their cleavage sites. NetSurf P3, which is protein secondary structure and relative solvent accessibility. DeepLock, which is for protein subcell localization in eukaryotes, and DeepLock Pro, which is the same in prokaryotes. NetSol-P is the prediction of solubility and usability of proteins heterologously expressed in E. coli. DeepTM-HMM for transmembrane segments in proteins, and NetStart for translation initiation sites in eukaryotes. I will talk mostly about Signal P6, and I will briefly mention NetSurf P3 and NetStart 2. And the others you will have to read, look up in the literature. Now, for Signal P, I will start by introducing the problem of protein sorting. There are sorting signals, you could call them zip codes, in the amino acid sequence. And in 1999, Günther Blobel was awarded the Nobel Prize for this discovery. Some of these sorting signals are called signal peptides, or leader peptides, or signal sequences. And the signal peptide is the signal for transport across the membrane of the ER or the plasma membrane in prokaryotes. And now the figure here is showing the eukaryotic situation, where proteins with signal peptides by default will continue from the ER through the Golgi to the cell membrane. So both in prokaryotes and eukaryotes, signal peptides are essentially signals for secretion. Well, what do they look like, these signal peptides? Well, they are N-terminal parts of the protein chain containing a hydrophobic region, also known as H region. And before the hydrophobic region, there's an N region, often containing positively charged side chains. And after that, there's a C region with more polar side chains. Positions -3 and -1 relative to the cleavage sites is normally occupied by small and neutral amino acids. This was just one example, so what do they look like in general? Here are some sequence logos where you can see the hydrophobic region. Maybe I can get a pointer. Yes. So you can see the hydrophobic region here. You can see the motif around the cleavage site here. And there's some information after the cleavage site also in bacteria, but not in eukaryotes. As you can see, there are similarities and differences between these three groups of organisms: eukaryotes, gram-negative bacteria, and gram-positive bacteria. And traditionally, prediction and analysis have been separated into these three groups. Then there are some, in prokaryotes, some special signal peptides. There are the TAT signal peptides. TAT stands for twin arginine translocation, and that's named after the two arginines in a motif which is contained within the N region of the signal peptide. They use a different translocation channel, and in contrast to proteins with normal signal peptides, these are transported in a fully folded conformation. Then there are lipoprotein signal peptides. They use a different signal peptidase, also known as signal peptidase 2 or lipoprotein signal peptidase, which attaches a lipid group to the mature protein, to this conserved cysteine in the +1 position. Then there are the type 4 pylene signal peptides that uses yet another signal peptidase 3, which cleaves before the hydrophobic region. Then when talking about signal P6, I have to compare to the baseline, which is the previous version, signal P5. Signal P5 was a deep learning architecture with two convolutional layers, and between those a double LSTM layer reading the sequence forward and backward. Then the output of the convolutional layer was sent to CRF, conditional random field, which would then decode the sequence. The news in signal P5 was, it was the first version that was based on deep learning, and it had this conditional random field. And instead of training separate networks for each organism groups, eukaryotes, gram-negative bacteria and gram-positive bacteria, we trained one network on all the groups and had an extra input unit inform the network about the origin of the sequences. This transfer learning made it possible for the first time also to predict signal peptides in archaea, where there are very few experimentally known examples. And signal P5 could differentiate between normal signal peptides and lipoprotein signal peptides, and also recognize the TAT signal peptides that I showed you on the previous slide. But there were some unsolved questions in signal P5. First of all, there are some proteins that are both TAT and lipo at the same time. Use the TAT translocon and the signal peptidase 2. Actually, we didn't know about this before the reviewers assigned by Nature Biotechnology made us aware that these proteins exist. There were also the signal peptides of type 4 pylenes, those cleaved by signal peptidase 3. And the question of the diversity of signal peptides, the gram-positive, gram-negative distinction in bacteria is not totally clear-cut, and how should we treat various bacterial phyla. And last but not least, the question of how do we make use of all the unannotated sequence data in the databases. Here's the size of SwissProt, the curated part of UniProt that has little more than 500,000 sequences. And this is where we find all the experimentally confirmed data that we need for training prediction methods. While in the unreviewed part of UniProt, we have more than 250 million sequences. And of course, these 250 million contain a lot of signal peptides, so how do we make use of those data? Protein language models is the answer. So as Brian showed you a moment ago, these are deep neural networks trained on unlabeled protein data. They are trained using the masked language model objective. So you mask out single amino acids and make the network predict which amino acid was at the masked position. They learn protein features from very large data sets, not necessarily having any labels, any annotations. And to use them in a downstream task, we then replace the language modeling head with a classifier. And pre-trained language models are publicly available. We chose to use the one called ProtBERT, which is described in this paper. So the signal peptide prediction model takes as input an amino acid sequence, sends it through ProtBERT, and which projects the amino acids to a context-dependent representation of a high dimension. They are then sent to a conditional random field which labels the proteins as signal peptides or non-signal peptides. I find it necessary to show you the size of the data set here, because when we for normal signal peptides, we have in the order of thousands of examples for eukaryotes and some 100 examples from the gram-positive and gram-negative bacteria. Numbers in parentheses, I should mention, those are after homology partitioning. No, that's before homology partitioning, and then this number is after homology partitioning. Note that some of these categories are really, really small. The TAT-SP2 signal peptides, the ones that we didn't know existed, we were only able to locate 30-something, 33 examples after homology partitioning in the databases. And try training any deep learning model on less than 40 examples. That is very hard, but for a language model, it's possible because the language model has already seen examples of this in its unlabeled database. So for the results, here I show you them as correlation coefficients. The blue bars here are the signal P5 performances, while the orange are the signal P6 performances. And as you can see, in some of the cases, performance was more or less equal for the large categories, but for the small categories, the signal P6 is way better than signal P5. In fact, for categories such as TAT-SP2, the performance by retrained signal P5 was too low to be of any practical use, but signal P6 with its language models could do it. For cleavage site precision, that is how many of the cleavage sites we predict are biologically real, there was improvement of all classes and organism groups, but again, especially for the rare classes. And here's an interesting experiment. As in signal P5, we had an extra input unit in signal P6 informing signal P6 which organism group the sequence came from, either eukaryote, gram-positive or gram-negative bacteria, or archaea. And if we shuffled the organism labels to give worthless random information in this unit, performance went down. You can see the orange bars are after shuffling the labels, the blue bars are with the label information. When we did that same experiment with signal P6, the differences totally disappeared. So signal P6 doesn't care about the organism group input information. This makes it applicable to metagenome data where we don't necessarily know the origin of the sequences, and it also makes it applicable to various bacterial phyla that are not necessarily well classified into the gram-positive, gram-negative divide. When we look at, again, signal P5 retrained and signal P6 performance on those sequences that were removed during homology partitioning, we can see that performance depends on how closely related the sequences are to the training set. But the performance drop when you go to lower percent identities is much worse for signal P5 than it is for signal P6. So signal P6 with a language model generalizes better to examples that are remote from the training set. A little bit about understanding, opening the black box a little bit. So we have used t-SNE to depict the representations in the language model before fine-tuning and after fine-tuning on the signal peptide task. And the color codes here show soluble proteins, transmembrane proteins, signal peptides, TAT signal peptides, and so on. As you can see, after fine-tuning, the groups are neatly separated, but even before fine-tuning, there's quite a neat separation, meaning that the language model has learned something about signal peptides and different classes of signal peptides already before fine-tuning in the pre-training. Now let me switch gears a little bit and talk about 1D descriptions of protein structure. That is secondary structure. It's relative solvent accessibility of each amino acid in a protein, and the 5 psi angles, the rotation angles across the bonds in the protein backbone. Those can be predicted from sequence, and we had a predictor called NetSurf P2 that was based on profile encoding of the amino acids. Well, it worked okay, but even with a high-speed profile generation such as MMSeq2, it would take a lot of time. In NetSurfP3, we replaced the profile encoding by a protein language model encoding, in this case ESM1B, and used the rest of the architecture was identical to NetSurfP2, and it turned out to have more or less the same performance in specificity and sensitivity, but it ran up to 700 times as fast. So instead of protein profile generation, which takes a long time, we just generate embeddings using the language model, which is a lot faster. Spot 1D LM, as you see here, is then our closest competitor, which runs around 5 times slower. And let me switch gears one more time to talk about another problem, namely the selection of the translation initiation site in eukaryotic messenger RNAs. There's this well-known scanning model of selection of the start codon, which says that the small subunit of the ribosome scans from the 5' part of the messenger until it reaches the start codon, which is almost always AUG, but it's not any old AUG. It's an AUG in a correct context. And this context is known as the Kozak consensus motif. In vertebrates, it's said to look like this. And it's not always the first AUG, which is the start codon. In more than 50% of human messenger RNAs, there's at least one AUG upstream of the annotated translation initiation site. This means that translation initiation site or start codon identification is a non-trivial task. And we made many years ago a method called NetStart for predicting the correct start codon. Now this has been updated to NetStart 2, which uses some local information around the AUG and some global information in a larger window. The local information is one-hot encoded and presented to a feed-forward layer here. And the global information is encoded by a protein language model. So we simply translate the sequence in the reading frame of the AUG upstream and downstream. And upstream, it's of course nonsense. This is not protein. Downstream, there is protein. And then we also have an input signifying the organism group. And that actually works. We get a much better prediction than NetStart 1. And we have shown by ablation studies that it's the protein language model that makes the main part of this improvement. So this was NetStart 2. To summarize, what can a language model do for protein science? Well, the trained language model has seen millions of unlabeled protein sequences, meaning that it has deep knowledge of how proteins are composed. The downstream task can then be trained with a small number of labeled proteins, and this means that this approach is less data hungry than other deep learning approaches. There's no need for profiles, multiple sequence alignments as input, and it can distinguish between real protein and nonsense translations. We have come to the acknowledgements. I want to acknowledge all these people, co-authors of the publications, and especially I want to mention Felix Teufel, who made many of the figures for this presentation. Finally, the availability, web servers here, papers, and my email address. Thank you.
We have time for a few questions.
Hi, pretty good talk. Actually, I'm not working on the AI field, or I'm just a biologist. I ask a naive question. So you find this subcellular peptide, localization peptide, is pretty striking and interesting. I'm wondering whether you can find those E3 diagram or E3 binding domains based on your study or based on the prediction. One could be used for those degrader or those molecular glue design for the drug design.
For the drug design, yes, I believe that can be done. It's of course always a problem. How do you get labeled data for training the downstream model? But
yeah.
I believe it could be done. Yes.
Yeah, I mean for this E3 ligase binding domain or recognized peptide in the protein, can you predict which, for example, one specific E3, like the cerebrum or VHL, which motif they can recognize? Or
yes.
Whether I'm not sure whether I explained clearly or not.
No, of course, I'm not an expert in that field, but I believe it could be done with the same approach that I'm using, yes.
Oh, it's approachable. Okay, thank you.
Thank you, Professor. It's a really nice talk. I have also a naive question that, do you think that there are any kind of undiscovered new signal peptide, and do you think that the current model can help you to discover them?
I have no reason to believe that there are undiscovered classes of signal peptides, but of course biology is always more complicated than we think. So never say never, right? But no, I have not seen any evidence for that.
Thank you.
Okay. If you don't have more questions, then let's thank Henrik for this wonderful talk. And okay. Let's welcome our next speaker, Dr. Noelia Ferruz, who's a group leader at the Center for Genomic Regulation in Barcelona. She'll be talking about protein design using language models.
Hopefully it doesn't fall over. There we go. Hello. Yeah. Thank you. Thank you very much for having me here. Thanks for the organizers. It's my first time also in China, so I'm double excited. And I want to tell you about how we are using and training protein language models to design new proteins. We dream of designing functional proteins because proteins are the most amazing machines we have on Earth. They carry nearly every function that we see in living beings. And like for example, from generating energy to signaling, and they do so with extreme efficiency. What we see here is a beautiful snapshot of a eukaryotic cell. And every colorful point that we see here is a protein that is working in orchestration in a very crowded environment. So we see microtubules there that are like highways for transport. And we also see those yellow cages. They are clathrin-coated vesicles. So because they are so efficient, nanomachines that work also in sustainable condition, it's no wonder that researchers for decades have been trying to design novel, completely new proteins that could carry as diverse and beautiful functions as these ones. However, as we possibly, many of us know, this has been tremendously challenging. It's been, for decades, really challenging to design not only proteins that were functional, but also proteins just that were physically done in vitro that could fold and present and be stable just in an in vitro solution without being farther and having even a function. What we see here in this plot is a very nice paper that came out recently. It's the Protein Design Archive. And it gives an overview of how proteins have been designed and deposited in the PDV over the last decades. And we see that the y-axis is how many proteins were designed per year. So we see that in the 90s, it was only a handful of proteins that were designed every new year, so from 5 to 10. Then things started to get better, so the community was able to get more proteins designed. In the 2010s, we see that this change in slope, and that's thanks to computational methods that allowed researchers to design proteins much more efficiently. These are methods like Rosetta that were based on physics-based purely energy functions. And that's because we know a lot of the physics of these systems, like we know the solvent interactions that they perform in their environment. We also have information of their 3D structures, so we can compute the distances and the van der Waals interactions. We also know we have information about their angles, so we can compute the dihedrals and some other forces. And all these can come together and come up into an energy function that we can optimize. So this is what led to the success in the 2010s and eventually also to the Nobel Prize recently. And even if it has been so challenging, I like to think that we are seeing an increase in slope in the recent years. The field of protein design has undergone a revolution, and we are seeing more and more proteins coming out per week. And often, not only they are structurally soluble, so that we can characterize them, but also they tend to often now the field is driving towards having proteins that also perform a function. And that has been powered by advances in AI. I like to think that AI, that the revolution that we are witnessing in the last 5 or so years comes from different information channels. The first one will be language. As we all know, there are many advances in NLP, so that in our daily lives, we use models like ChatGPT or DeepSeek. We also are seeing a revolution in computer vision, and we are seeing a revolution in video generation. And the way that the protein research field has undergone this revolution is because proteins, they are inherently well represented in these 3 information channels. Like, we don't really need to do much from the architectural side point of view to just take the proteins and train them on the models that have been very successful in NLP and computer vision. And this is for just the way that proteins can be so flexibly represented. We can represent proteins as language. If you look at a protein, it's basically, it encodes a language. So proteins are a selection, a string of amino acids that is, in a sense, the same as an alphabet that we will have in human languages, like in English. And then these amino acids, they combine together to form secondary elements, the same way that that letters form words. Then these words form sentences that carry a meaning, the same way that these elements combine to form domains that have a function. And we also have sentences that combine into paragraphs and longer text and books and so on that carry longer meanings, are coherent. The same way that we have domains that combine into quaternary structures that interact with other elements in their environment. And this is for language, but proteins also have a structure. So it's also no wonder that they have been so successful in the training of models that are diffusion-based, like DALE3 and so on. But because if we think of a protein, it has atoms in space with X, Y, and Z coordinates, which is literally a 3D matrix, where for every atom we will have the three coordinates. And that's literally what a picture is. So it's a picture is a representation of three channels, like red, green, and blue, and that gets represented into pixels. So literally, a picture is the same as a protein. We don't need to do much changes in the architecture of models like DALE3 to, instead of pass images, just literally start passing protein PDBs. And also, we are not seeing a lot of advances in the field, but there has been in computer vision lots of advances as well in the generation of videos. We see a lot of scary sometimes deepfakes. And proteins, we tend to forget that they are dynamic, but we have a lot of data for dynamics as well for proteins that we could take into these models literally and start generating protein trajectories as well. So I expect intense research in the short future in time series as well. But we've been focusing on language because, in my opinion, a lot of beyond so the function, but also, as we know from the dogma biology and some of the things that Brian was saying earlier this morning, so this language encodes the structure. It also is going to encode the dynamics of a protein and eventually also its function. So in my head, this language, this protein encodes a grammar that hopefully with models that arose in the field of NLP that we can learn but also speak. And that's how we started training protein language models. Our original goal was to take the bulk of huge databases of protein sequences and train models on them so that we could get these models to speak protein and teach some of these biological principles for us, but also use them to design proteins that don't currently exist on Earth. So this is what we do. We've seen the transformers several times now in the talks in the last 2 days, but I thought I will still give you a short overview of how it works. The original transformer is an architecture that came out in 2017, and it was designed by Google. That at the time they were trying to improve Google Translate. Maybe you don't remember, but back then Google Translate was actually pretty bad. We will have not so good translations. What they come up with is a bimodular architecture that has an encoder that takes as input language, so it could be a sentence in English, and then through an attention mechanism is going to output that, pass that information to a decoder that is going to produce an answer, could be the translation in Chinese. And then this worked extremely well for a fraction of the compute of previous models. So it's no wonder that it started a revolution in this field, but also in any other. It has also led to advances like AlphaFold and the genomic language models that we were seeing at the very beginning today. People also started not only having architectures that have the two modules, but also looking at the modules independently. So instead of training an encoder-decoder, we could also train a decoder, and that literally is an architecture that is going to learn how to generate the next token. It does so by learning the probability distribution of the individual sentences or the proteins by splitting them into tokens that could be the single amino acid. So it learns to speak one at a time, and it learns the probability of what is the next token based on the previous ones. This is more or less what the attention mechanism is doing. So when we do so, when a model has learned over millions and millions and millions of proteins that we have on Earth, when we give it a single amino acid as input, it's going to be able to be generating proteins that are feasible and that represent the training distribution that we have on Earth, but that are completely new. So they have learned how to speak the protein language. The way that this works more technically is we need to start always with a big database of protein sequences. This is the way that I trained ProGPT-2 some years ago, no, 3 years ago. And then for that, at the time, I took UniRef that had almost 50 million sequences. Then I split them into chunks, I tokenized them. And then that was passed into 128 GPUs for a few days. That led me to ProGPT-2 that had 36 layers and over 600 million parameters. This was a proof of concept for us. And we could observe that indeed the model is able to speak proteins, and it speaks many proteins very quickly. You could let it speak for overnight, and then the morning after you have a good bunch of million proteins there for you to analyze. Usually in English, we will just look at the text, or in Chinese, and then we will see whether it's grammatically correct, whether it makes sense. But here we have the problem is that I look for at proteins every day in my life several times, and I still cannot tell if a protein is going to be foldable or not, unless I see something overly obvious like a repetition of alanines. So it's very hard for us to try to understand this language. Thankfully, we have now these models that speak it very well for us. So the first thing I did when I trained this model is to check whether the proteins will make any sense. So first I look at whether they will be well folded according to prediction tools like AlphaFold, and where will they be in the protein space. So what we see here in this network is a representation of the protein space, and I colored every protein is a node, every protein is a point in here, and I color them depending on the topology, so the shape that they have in solution. So we have all these categories in here. So a protein could be all alpha, or all beta, or alpha beta, alpha/beta, and so on. And what I observe is that mostly the proteins that are generated by the model that are represented in white, they tend to expand clusters that are already known in nature, but also occasionally from time to time, they will also start a new cluster. So the protein that we see there, 4266, is a protein that was completely novel. It didn't match any topology previously known in databases, and it has a very low sequence identity to any others. But most of the time, we will see proteins that are homology-based, like they explore clusters in new points in clusters that were previously known in the training set. However, what we most like from this back at the time, that we were most happy with, is that the proteins that the model generate are very nature-like looking. So in protein design back in 2021-22, when we were working for the first time with this, most of the designs that we had been able to do, they were very idealized with minimal loops, short elements, no embodiments, and they tended to be very rigid. And here we were able for the first time to see designs that were diverse and very far away from what's known in nature, but that they had loops and embodiments, and they could have the potential for flexibility and eventually also bind partners or perform enzymatic functions. So this was very reassuring, and then we went to the lab and tested all of these and many others, but I'm not going to show all those specific results today. I'm going to show you some of the later models that we have in training. Even first, we trained ProGPT-2, and we were happy to see that it generates proteins in unseen regions of the protein space. Often, we just don't want to generate a random protein into the space. So often, we have an application in mind, like we may want to engineer proteins for different reasons. So we trained sign control, which instead of being a completely unsupervised model, you as a user can input what is a specific chemical reaction as a functional tag. Like we can input, give me a serine kinase or a carbonic anhydrase, and it's going to only output sequences for that specific label. And then very recently, we've trained RegSign, which instead of taking a label as such, like carbonic anhydrase, we directly can input also the chemical reaction so that we also have control over the substrates and products. Briefly about sign control, we've been testing it thoroughly among many targets. So I will show you some of them, but first let's look at how was this more or less trained. Because there were some interesting bits. We did this in collaboration with Basecamp Research, which is a company based in the UK. And then we observed that in public databases, we have a very unbalanced situation of protein sequences. So this plotting here shows how many proteins do we have for every easy label. And easy label is a way to capture what is the enzymatic activity that an enzyme is performing. So it's a very hierarchical way of representing protein activities. And then we see that for some of the labels, we have a lot of data, and for some other labels, we practically know nothing. Like there's more than 50% of the members in here in this data set that only have, for one is a label that only has one protein. So this makes a very unbalanced for a data set for training, for which we decided to tokenize the labels as well. So when we pass to the model the protein sequence, we also pass the label, like in there, 2716 and 2712. So the model is able to infer that the labels, because they are hierarchical, that these two sequences, they share something in common. In here, what we see is, the two sequences, they share a 2, so they are oxidases, and then they are more similar the more we go to the right. So in these cases, they are oxidases for sugars, and the only thing they differ is in the substrate specificity. So the first one is glucose, and the second one galactose. So the model, even if it has seen a lot more sequences for the glucose family, it can also infer some of this information, translate that, transfer this knowledge to the family of the galactose oxidases as well. After training, then we only need to input the model, the easy label that we want, and it's going to generate some sequences for that specific easy label. So after training, we can use it in zero-shot for the generation of specific enzyme families. And we did so for many families, but also we were happy to see that often we can also take the model and train it on, fine-tune it on other cases that are not enzyme families, and it will also perform well. So we've been designing alpha amylases, triphosphate isomerases, thin fingers, and other many enzymes. But today I will only focus briefly in the first three because it's for the sake of time, but also because I want to show you how we can optimize the models beyond what is in the training set. This case that I particularly like is the design of lactate dehydrogenases. In here, what we did is to fine-tune the model specifically on and only on lactate dehydrogenases that have been collected in remote places. And then after that, we generated several sequences. We did not even filter them much. And 20 of them were tested, as we see here. And we were happy to see that many of them were performing better in terms of activity than the controls. We took 2 of them and lyophilized them. So Sylvia took them and made them powder. And then this powder, we were able to put it into catalytic pots that we see here. So our designs, the LDHs, were inserted into the catalytic cascades, as we see here. And we were happy to see that after overnight, we had some preliminary conversion. This was particularly exciting because we had not filtered or optimized those sequences. So that's just LDH19 that had been outputted by the model, and we took it, literally did nothing, no engineering, made it powder, and put it into this catalytic one pot. And we already observed some preliminary conversion without doing any further engineering or directed evolution. So that was very exciting to see. And then we did many other families at that time, but also one of the ones that I particularly like is that we tested alpha amylases, because previously we had been always testing 10 to 20 designs maybe. So we never have enough numbers to see some statistics and apparent performance. And for the alpha amylases, we were lucky to see that we were lucky to test 200 at a time. And we saw that many of the designs, activity is relative to the wild type. So when we see that plot, the higher the better. And we see that for many designs, we were having activities that were better than the starting point, than the wild type. However, when I paid a much closer look into the data, if we look at the plot to the left, we see that the activity of the training set also had some cases that were better than the E. coli wild type. This means that the model had been producing active sequences, but also because there were very good active sequences in the training set. And then I wonder about, well, how much can the model really be pushed beyond what is in the training set? And does this happen also for any other properties? And indeed, that was the case. We checked for any property under the sun for many others. And then we observed that whenever we see this distribution in gray, the distribution in gray is the property for the training set, and in blue is the same property, but for the same number of generated sequences from the model. And then we see that the distribution for these 2 cases, but also many others, they tend to be very well overlapped. So the length, if the model has seen sequences around 200 to 300 amino acids, it's going to generate also sequences in that range. And the same for the predicted structure, predicted properties like the PLDTT from ESM-Fold. This poses a problem because often, if we go back here or there, we may want to, when we want to have sequences that are active, we may want to go to the level of, we want to engineer them to the level of an activity of 10 or 12, and how do we push the model there if it's only anecdotically going to be sampling from those faraway regions? So we've been doing this with several methods. 2 of them that we have been working on lately is reinforcement learning, but also causal interventions. I will present more the first, but let me give you an overview of how they differ. In reinforcement learning, we have an agent that is the model itself, the protein language model. The model is capable of generating sequences. Then we are going to have an environment or an oracle that could be as simple as AlphaFold that is going to give us a value, a score of how well the protein is performing in a certain context. And then we can take that value and put it back to the model. So we can tell the model, you know, those sequences were actually not working very well, or oh, you are doing well in those sequences. It's kind of like the same way that we learn when we are kids and adults by interacting with our environment. So by doing these cycles, the model gets better and better over time. Whereas causal interventions, what we are doing here is to train the sparse autoencoders. So we train models, these classic models from mechanistic interpretability that Brian was also showing earlier today. We have an internal representation of the model that could be the residual stream, and then we pass it to an encoder-decoder architecture that is going to try to get a representation that, instead of like capturing a lot of information into a single small vector that we call often that is polysemantic, it's gonna get a much larger vector that we say often is monosemantic, so that it's able to decouple a lot of information into specific single features. And some of these features, we are going to be able to see that they actually translate into what the model has learned about activity. Some of these features only fire for active sequences, and some of them will only fire for extremely active sequences. So we can take the model and intervene the model and only maximize those that are the most active ones. And we see this working very well at a single shot instead of like doing several rounds of evolution. But I will focus on the first method today. This is more or less what reinforcement learning does, at least in my mind. In traditional protein engineering, we used to do little steps. We would start with some of the proteins in there, some of the points in orange, and then we do little modifications up to sort of like a hill climbing algorithm until we reach the closest maxima. And this worked very well. As we know, it was even granted the Nobel Prize in 2018 to Frances Arnold. And the way the PLMs work, the protein language model works, is like maybe they are sampling much larger spaces of the same family, but however, they are always going to they are bounded to the training distribution. So only anecdotally, maybe they will be sampling from the very high peaks, but often they are going to be sampling whatever we had in the basins. And what reinforcement learning does is like through rounds of iteration, it's going to get to the top. This is called agentic learning or agentic workflows. And in short, even if it sounds incredibly fancy, agents are nothing else than a system that is going to be learning from interacting by an environment. And the simplest agent we can think of is a thermostat, because it's going to sense temperature, or it's going to think whether it's cold or hot. It's going to have a threshold, and then it's going to accordingly act and improve the temperature regarding, so in regard to what it has seen. And more technically, so we have a model like sign control that it takes as input an easy label. So that for example, we will be saying, oh, we want some carbonic anhydrases. Then the model generates some carbonic anhydrases. Then we go to the lab with those. And some of them are going to be working better than others. And we can tell that the model back. So we can say, you know, some of them were actually working pretty badly, and some others are good. And then the model is going to readjust its weights and produce a second batch that we can test as well. So this is what we implemented. We implemented a method called ProdRL. So we now encompasses several reinforcement learning algorithms like DPO, direct preference optimization, but also GRPO, which is the way that DeepSeek was trained. And then first we did several trials in silico, from which I'm only showing one. When, for example, we have a normal PLM like sign control, it will just generate again from what it has seen in the training distribution. So it will mostly produce beta carbonic anhydrases versus alpha carbonic anhydrases for that label, simply because there are more beta than alpha in the training set. But what if we want to go from beta to alpha? Then we need to tell the model, well, whenever it generates a beta carbonic anhydrase, we need to punish it. Whenever it generates an alpha carbonic anhydrase, we need to reward that. And that's what we did we show in the plot there. Over several rounds of iterations, the model goes from generating only like 10% of alpha carbonic anhydrases to up to 100 almost after 5 iterations. So by punishing whenever it does wrong and rewarding whenever it does good, we are able to see that we can drive effectively the model from beta to alpha. And we thought, well, maybe it's doing that because it's copying over the same sequences over and over. But it actually, we look at the diversity of generated sequences over the iterations, and we see that they didn't decrease the diversity. So we see the similar number of clusters from the beginning to the end. So it does generate, it's able to drive the model to new regions of that space without, and not at the cost of the diversity of the generated sequences. So we were very, very happy to see that. And then we went to the lab, and some of the things we've been testing are the binding of binders, for example, for the EGFR receptor. So we first did an iteration in which we had some nanomolar binders in the long range, almost micromolar. Then we told the model with this and other papers that were in the literature, you know, some of these sequences were good, but you could do better. So we produced a second round. And then this second round, we had low nanomolar binders. And then we did the same. So we told the model, we gave this feedback to the model, and it produced a third round. And in this third round, we have mostly low nanomolar binders. So in 3 rounds, we were able to see a decrease of the activity of the KD values. I should remark, in case you are not familiar with the notation, the lower, the better. So we want to push the model towards generating lower and lower KDs over iteration. We want to optimize how tight the binding of the new proteins is. We can also, as very similar to what Brian showed, we can also look at the interpretability of these models. So what we can do is to exactly like Anthropic did for cloud, which is what Brian also showed today, we can detect what the model is looking at for each of those generated sequences. So we are working a lot into the explainability of this model. So why is the model is getting better? And if the model is able to generate proteins that are functional and so well performing, can we actually learn something from it? Can we extract why is it doing this? And so some of the things that we can do is to decouple the reward into the specific residues, so we can see where the model is assigning more reward. And sometimes some of those residues are not readily evident for us. Like some of those residues will be far away from catalytic pockets, or in here from the interaction area to the receptor. And very briefly, and just because for the sake of time, I will only present the very last results we have for Rexime, which is our most controllable model, so that we can directly input chemical reactions in the form of smiles, and it's going to give us as output a dilord enzyme for those chemical reactions. It has several advantages, and is that we are no longer bounded to the training set, so we are no longer bounded to the chemical reactions that we had originally in the training set, but it has also learned to translate between chemistry and biology. Our hope was that even if the data is scarce, we see models, or we see Google Translate doing so well between languages as far away as English versus Chinese, that why cannot maybe we dream about translating chemistry to biology and the other way around? So we've been doing extensive work lately trying to see how the model generalizes and how well it performs. We've been looking at the generalization of these models. Previous models, we were not able to generalize beyond 0. So what we see in the plot is the fingerprint distance. That means how far away the sequence that we input to the model is to any other reaction in the training set. So like for chemical reactions that are very far away, how much the model performs. So the y-axis will be a measure of performance. And we have several cases for which this works very well. So we have chemical reactions that the model has never seen, but still it outputs a protein that we know is feasible and good. So we went to the lab and tested formatic hydrogenases. These are enzymes that are particularly attractive because they often have been, so the nature has optimized them to go from formic acid to carbon dioxide, so in the oxidation side, but we could also, it has also been observed that some of them are able to reduce, so we can do CO2 reduction, which will be extremely cool because we want to capture CO2 and then transform that to valuable chemicals. So we took the model and trained that on formatic hydrogenases through reinforcement learning. So we took all the data that we found in every paper on Earth about formatic hydrogenases and told the model the Kcat values, the Km values, and every enzymatic parameter that we were able to find. And then the model generated new formatic hydrogenases that were aligned with the data. And then we only freshly, last week, we were able to get these 20 designs. The 20 of them that we generated work well. They were expressed and purified. And we have 7 now, as of this week, that had improved activity, and the rest were with similar levels to the wild type. So we were very, very happy to see. And only lastly, so we dream of like not only incorporating chemical reactions and other modalities, but also dynamics, as I mentioned before, structures and also physics, because we tend to overlook them. And then we are coupling all these agents with robots, so that our dream is that the agents are able to autonomously generate the sequences, so that they can test them autonomously in the robot, that they are going to get the feedback back and improve over time. And we dream of also getting insights, biological insights into why the models are doing so well, what can we learn from them if we still cannot generate design sequences rationally after years and decades of research. With this, I want to thank my team and also the funding agencies for allowing us to follow our dream, and also the many collaborators, and you again for having me. And I'm happy to take any questions. Sorry for the delay.
Hi, Nolia. Thanks for your fantastic presentation. And talk you discuss about the ProGPT Zyme control that takes fine-tuned controllable generation, reinforcement learning for guided generation. And that's the beginning of your talk. You show one cool analogy between language models between the protein language. So my question is that in large language models, sometimes they have some robustness issues, some security issues, such as jailbreak attacks. Have you ever witnessed or foresee the potential dual-use risks in the protein language models? And how can we prevent such issues? Yeah. Thank you.
Excellent question. We are looking a lot into this part of explainability as well. So how can we jailbreak, or what are adversarial attacks for these models? For our PLMs themselves, because they often have no input other than the labels, it will be hard to have adversarial attacks. But in the community, it's been discussed how do we prevent the generation of viral proteins, for example, and so on. And the traditional technique, in the last 2 years, maybe what people have done is to remove those from the training set, or just like have a statement in to prevent this misuse. But we're going to need in the future ways to, like, for example, mark those sequences, or to label the sequences that are generated by the models, so that we can trace them back whenever something happens. But yes, a super interesting question. We are also looking into this, but exactly have no solutions at the moment.
Yes, thank you for your answer and presentation. Thank you.
Thank you.
Do we have any more questions? Okay. One, I think, last question in the interest of time.
Thank you so much for sharing your presentation with us. So I have a question. You mentioned that ProGPT-2, it creates the protein sequence that are naturally like protein, right? Like the sequences. So what kind of syntax it takes from a protein to create that kind of proteins that look like natural protein, and how do you evaluate the biological plausibility of those created proteins?
For that model, so for the first question is like, you ask exactly what kind of, like, how do we evaluate whether there are like natural sequences?
Yeah.
By topology.
Yeah, like the grammar it takes from a protein to create that naturally like proteins.
Yeah, those, like, exactly what it promotes. For ProGPT-2, we didn't do much more. It was more of a proof of concept. But what it takes the model does to generate something specifically is still a big mystery. But it tends to sample from clusters in the latent space that it has well learned. We can check that always explicitly after training. So we can check by topology measurements or like sequence identity where it does, like, where is it taking this information from. It's much more like interesting that these models, like, they produce, that we can infer the function directly. Diffusion models, somehow they generalize a lot better. They can easily go to the 0% sequence identity. However, designing function with those is like, they require a much more inductive biases from the user. So it's, yeah, I hope this answers more or less your question.
Okay. I think in the interest of time, we'll limit the questions. Maybe you can ask your question in the coffee break. We don't have a lot of time for coffee break, so I think we can start at 10:35, but promptly at 10:35. So like about 18 minutes for the coffee break. And let's thank our speakers from the first session again. Thank you.
In this era, what are the missions of a university?